I'd like to welcome all of you to the 26th Annual Raymond Berry Memorial Lecture. And before we get started, I'd like to introduce Dr. Berry's daughter, Miss Dorothy McLemore, who's there. And Miss McLemore has a sister, Margaret Thompson, and Margaret and Carl Thompson's daughter is here, Alicia Smith, so welcome. So this Berry lecture got started really with Dr. Berry. Once upon a time managed cattle for the 9 Bar Ranch, which is was located at that time near Hempstead, and so the Wortham Foundation gave an endowment to get this started, and people have added to it since that time. But that allowed us to have this every year, and so it's great that we're now in our 26th year, and looking forward to next year. I think this year with the COVID lapse, attendance is down a little bit, but I think we'll get back to normal probably next year. So today we're going to ask Dr. Bill Foxworth, who's here at Prairie A&M, to give an introduction to Dr. Berry.
Well, Dr. Berry was a member of the faculty at Texas A&M University for many years, contributed to the establishment of the discipline of reproductive immunology through pioneer studies using embryo transfer to evaluate genetic factors affecting reproduction. Excuse my voice. So the Raymond O. Berry Memorial Lecture is presented annually by a distinguished scholar, today Dr. Johnson, in the field of reproductive immunology on issues of importance to animal agriculture, veterinary medicine, and human medicine. Looking at Dr. Berry's biographical sketch, and I really appreciate that that this is a trainee. There are a lot of trainees here, okay, because this really, this history is very important to understand. Dr. Berry was born in Chico, Texas on October 28, 1902. His BS from North Texas State Teachers College, 1929. His MS from the Agriculture and Mechanical College of Texas, pre-TAMU, 1932. His PhD in cytology at Johns Hopkins University in 1939. I believe that's about the same time that Dr. Burkhart was around there. And he has associate geneticist. Then he became the associate geneticist and associate professor of physiology at Texas A&M University, 1938 to 1952. He was a professor of animal husbandry, Texas A&M University from 1952 to 1960. He was the director of the Wortham Research Laboratory that Dr. Baser just mentioned in Cypress, Texas, 1960 to 1970. And then he was a professor of, he retired, but he came out of retirement basically, and was a professor of reproductive physiology and a director of family planning services here at Prairie View A&M University from 1970 to 1976. Dr. Berry was retired as professor emeritus from Texas A&M University, and he resided nearby in Hempstead, Texas, until his death in 1992. And bringing up the history right here, Dr. Berry was married to Grace Berry and had 2 daughters, Dorothy McLemore and Margaret Thompson of Houston, Texas. My mentor, Dr. Kramer, was mentored by Dr. Berry and was very close. They were both very close and is attending a family graduation here. I believe this is at Texas A&M, isn't it? I believe this graduation was at Texas A&M, was it? Yes. So talking about that history, I find this fascinating right here, okay? This is from his thesis. And this is the design of a stir that he had to make to be able to mix his samples, okay? This was 89 years ago. And the reason I say it's great to have all the trainees here, think about the equipment we have today, right? How many of us have to make our stirs? You know, so he was beginning to develop this field with, it's like working with stone tools, okay? And yet now we have all these great things, all the omics where we're drinking from a fire hose, right? And yet, this is how it started off. At Texas A&M, probably the seminal work that I'd like to refer to is presented here in the Proceedings of the National Egg Transfer Breeding Conference, 1949, in San Antonio. And his work was developing intergeneric and intraspecific embryo transfer between sheep and goats with Dr. Warwick. And here they are together. I'm going to assume they're discussing science at this point. But again, in the evolution, I remember just a couple of years ago at the TFRS meeting that was held in College Station when we were honoring Dr. Baser, and all of his trainees were there. They were showing lots of photographs. Now, in his discussions of science, I believe they were paddling in rivers in the Florida Everglades to discuss their science. So I don't think they were just sitting out on the porch discussing science. But together, this is very confusing. Every time I look at this, those letters are backwards, okay? So A is actually E, and E is actually A. They're in reverse for what's below. But the important thing there is that that I'd like to highlight is the A, which is actually the E on the right, is a sheep egg fertilized by goat sperm. Note the small abortive cell. No normal cleavage was observed. Did we all not just see all these beautiful photos today, right? The incredible photos that everybody has of their work with all the immunofluorescence and everything else. And yet, this is the state of the art. And this is what they were having to work with at the time to do this. And it just fascinates me. So in that study, the interspecific transfers that were done, the sheep with a goat recipient, they did 26. They had 3 living embryos that they were able to observe when they killed or they collected the females. They had 2 resorbing embryos, and they had no goat, obviously, to parturition. And then the other way, with the goat going into the sheep, again, you see they had no living embryos. They had 1 non-gravid, and then the 2 resorbing embryos. These are the photographs of the 30-day embryos. From the left is the goat, the center is the hybrid, and the right is the sheep. And you can see the goat and the sheep still in the condition that they are there. Still look fairly normal, but the hybrid is beginning to not look well. The 44-day embryos, again, the goat and the sheep, the hybrid is not developing. And this is 45-day embryos of the hybrid, the goat and the sheep. Now, these are not the interspecific transfer, but this is hybridization between the goat and the sheep. These are the goat and sheep hybrid embryos aborted at 79.85 and 143 days after breeding. These are the auto transfers that they did at the same time. The kid from the auto transfers. The lambs that they also had auto transfer from the 8 cells. These are obviously very early on in the embryo transfer field, folks. Newborn kid with its uterine foster mother, 8 cell. Again, lamb with its uterine foster. So these were studies to look at auto transfer versus transferring into a non-related recipient. So interspecies transfers. The sheep embryos appeared to have survived longer in the goats than the goat embryos survived in the sheep. That was the takeaway. So if you look at the sheep embryos from the goat foster uterine mothers here. And I was looking, every time I saw everybody's slides with the filamentous images up there that were just fantastic. This is what they were working with, obviously. So these were the goat embryos that were transferred into sheep, and obviously they had almost no development. So then another one of his major works when at Texas A&M was the cytological study of the maturation process of the ovum of the ewe during normal and induced ovulation. And this is the work that was published. It was very hard to get this one to be able to copy it, but you can see again the quality and what he was having to work with. And I really respect that. That would have been very hard to do. So then he moved on to the Wortham Foundation, as Dr. Baser mentioned, and they held an annual symposium on fertility of cattle there. And I picked out two highlights. This would be in 1963, the program, and what they did was they brought in people that were cutting edge at the time to discuss the fertility in cattle. And I'm just highlighting here methods of improving the conception rates in artificial breeding in cattle. This is in 1963. They were just beginning to move along with the artificial insemination in cattle. Not that it wasn't being done, but the technologies, the freezing of the semen, whether you could transport semen, whether they were using liquid nitrogen yet or just CO2. A lot of these things is what they were beginning to look at. The one that I really enjoy, though, is the 9th Annual Symposium, which was in 1969. And if you will look at this paper right here, a new method, straw freezing of semen. So having worked with a number of bull studs, I know some of us are still old enough to have worked with ampules and pellets on CO2. The ampules blow up in your hand sometimes, and the pellets were always hard to work with because you had to get them out and put them in tubes and thaw them and everything. This is a major step forward in the world of artificial insemination. It's also about the time that liquid nitrogen was beginning to be used for freezing semen, which that meant we could now start to ship this stuff around the world because we couldn't before because it was on CO2. So this is where they were working on cutting edge right here, very local to us here. This is just a note that the 1932 year of the goat embryo transfers and the 1933 year of the sheep embryo transfers. Now I mentioned when I mentioned that was 89 years ago for that tool. It's just kind of everybody take a thought right here. Somebody 89 years from now might be looking at your thesis, and they're looking at that Illumina, sequencer that you use or whatever, and they're thinking, God, that's an antique right there, right? So, jump. So, what is one of the sort of pinnacles of your profession? And that's being recognized as a pioneer of your field. And this is Dr. Kramer presenting Dr. Berry with the Embryo Transfer Society's, the International Embryo Transfer Society's Pioneer Award. And I know that he had asked for Dr. Kramer to present it to him also. And I hope Dr. Kramer's watching us right now, by the way. Excuse me. All right, I'm going to relate one story to you. I'm glad that there's people that weren't here 2 years ago for this, though. So in 1985, Dr. Scott Coonrod, and he wasn't Dr. Scott Coonrod at the time, he was Scott Coonrod. He had gone to Wyoming, to the University of Wyoming, to present a demonstration on non-surgical embryo collection in sheep. University of Wyoming is a big sheep university. That technique had recently been developed in Dr. Kramer's lab, because it's very difficult to collect, it was all surgical collection from the sheep, and this was the first technique to actually do it non-surgically. It's a little bit difficult, so there was a senior undergraduate student there that spent 3/4 of the day holding sheep legs all day long while he just demonstrated this to different groups. And the two of them discussed the work being conducted in Dr. Kramer's lab in interspecies embryo transfer, which was directly related to Dr. Kramer's work and being mentored by Dr. Berry. So there was a seed planted in that student's brain that day because he was interested in it also. But he was committed to going to college in Tokyo, Japan right after he graduated. But he was determined to come back and get into Dr. Kramer's lab. So in 1987, the student joined Dr. Kramer's lab group and worked on interspecific embryo transfer between the mouse and the gerbil. There was a technique that had recently been used. He had used it to make chimeric embryos, where you took the intercell mass from one species and you injected it into a trivectodermal vesicle of the second species to then transfer and try to overcome the problems of interspecies embryo transfer. That technique the reason he used that technique was because it had been recently used successfully to overcome the failure of interspecific embryo transfer between sheep and goats. So Dr. Berry's work, all the way back in the early 60s, had finally been overcome in the 1980s by Terry Roth and Anderson and Bondurant and Passion out at UC Davis. So the foundation for all this work that we're talking about was that seminal study that Dr. Berry did in 1963. Well, and before that also. Just so you know, that's my dissertation right there. The usefulness of diploid tetraploid embryo aggregation for facilitating xenogenetic embryo transfer between mus musculus embryo and unguiculatus, which is the gerbil. So my dissertation and all my work is based on that study, came off of that study and the trail that has followed it. So as trainees, I know, you mentioned today earlier, 400 and some odd papers, right? There's a lot of trainees that are on those papers, but I guarantee you, you'll have a lot more papers over the years, but everybody's going to have this family tree where Dr. Bazer's at the top of that, or Dr. Burkhardt is at the top of it, Dr. Johnson. It's going to carry on, and you're going to pass that on to your trainees too. So that's my presentation for the history of Dr. Berry, and I'm going to turn it over to...
So it's my pleasure now to introduce Dr. Greg Johnson, who's a close friend and colleague for many years, as I mentioned earlier, to present this year's Raymond O'Berry Lecture. Dr. Johnson is a native of Wyoming, comes from a wonderful town. I love the name of Ten Sleep, Wyoming. And so he got his BS, Master's, and PhD at the University of Wyoming. And his Master's and PhD degrees were in microbiology and reproductive biology, respectively. Then we were able to get him to come to Texas A&M, where I was fortunate enough to have him work in my lab for 2 years as a postdoctoral fellow. And then he was recruited to go to the University of Idaho as an assistant professor, and he served there between 2000 and 2002. And then we were able to recruit him back here as an assistant professor in 2002 in the Department of Veterinary Integrative Sciences, where he's risen through the ranks and now as a professor in that department. Dr. Johnson's received a lot of honors, so I mentioned a few of those. One of the most recent ones, he was named an Edges Fellow, which is from the Chancellor's Office in 2020, which will help him support graduate education and research. He's also received the College of Veterinary Medicine and Biomedical Sciences Honors Convocation Faculty Award for Research and Outstanding Mentorship of Graduate Students and Postdoctoral Research Associates. He received that in both 2014 and 2016. He's received the Gamma Sigma Delta Award of Merit for Teaching, and he's also received the Vice Chancellor's Award for Excellence and Diversity in Bridges to the Doctorate in Reproductive Biology Leadership Team. He's also received the Vice Chancellor's Award for Team Research in Uterine Biology and Pregnancy. Dr. Johnson also has received the Texas A&M Former Students Award at the college level for teaching and also at the university level, and he's also received at the university level for his research. Dr. Johnson is Associate Editor for the Journal of Reproduction between 2018 and present, and he's also a member of the editorial board of the journals Placenta and Domestic Animal Endocrinology, and he's also served on the editorial boards for Biology Reproduction, and reproduction over the course of the last number of years. He's also a member of the Society for the Study of Reproduction, the Endocrine Society, and the International Federation of Placenta Associates. And for the SSR, he served as a member of the board of directors for that society in recent years. So for all of us who have been involved in the Texas A&M for the last few years, we know that Dr. Johnson has been a key player in terms of the activities of the Interdisciplinary Faculty of Reproductive Biology. He's been an organizer of this meeting. We have the Texas Forum on Reproductive Sciences in Houston every year. And he's also very active in terms of being on the executive committee and organizing the meetings there. He's also commended for having mentored 41 graduate students. He's a member of 93 other committees for graduate student education. He's mentored international undergraduate students, 54 undergraduate Aggies, and 5 postdoctoral fellows. And his research has been supported pretty much continuously through grants from the NIH and from the USDA over the time that he's been at A&M. So out of that research, discovery research, he's presented 51 invited presentations at national and international meetings, 176 papers in refereed scientific journals, 13 book chapters, 26 illustrative drawings. So some of you, if you were paying attention, noticed that his name was under most of the drawings that came from the group at A&M. And he's also had his name on 170 abstracts presented at papers at scientific meetings nationally and internationally. So it's my pleasure now to ask Dr. Johnson to come in and present the 26th Raymond O. Berry Memorial Lecture titled Using Livestock to Understand the Immunology of Pregnancy.
Always beware of me and technology. Always. Okay, well, thank you very much, Dr. Baser, for that very nice introduction. All I can say about all that is I reiterate with what Dr. Baser said earlier this morning at the beginning of the meeting: surround yourself with good people. I've been really, really fortunate to be a long-term friend and a long-term collaborator with Fuller Baser, Bob Burkhart, Guy Yao Wu, and Hewan So. You know, we all know these people as talented people. We know these are hardworking people. We know these are generous scientists. So that's my advice to all trainees. Surround yourself with good people, and you rise with, I'm the bottom of that bunch, for sure. And the story about my hometown of Ten Sleep, Wyoming. It's named after a petroglyph in a cave. It's a teepee with 2 hands, and it's just that the Indian tribe, I think it was the Cheyenne, would move from Casper, Wyoming, up to Bridger, Montana, and back through the seasons. And they called their days sleeps. And it was 6 sleeps from Casper to Ten Sleep, and 6 sleeps from Ten Sleep to Bridger. So that's why we're called Ten Sleep, Wyoming. And if you go over the Bighorn Mountains, which is a big mountain pass, and drop into Ten Sleep, almost every tourist stops and gets their picture in front of that sign with the 10 sleeps. Mostly because it's the first mountain pass they hit on their way to Yellowstone Park, and they're pretty much spooked to death after coming off that pass because it's the steepest thing they've been on. So I thought today that it would be appropriate to begin with showing all the pictures of the past Berry lecturers. It's quite an illustrious group. My first Berry lecture was with Chuck Wira. I saw his talk. So I didn't get to see Tom Gill and Janet Rassant and Ann Croy's talks. And then I'd missed a couple more talks from Doug Ansack and Steve Witkin because, as Dr. Baser said, I went to the University of Idaho as an assistant professor for a couple of years. But I've been here for all the rest of these presentations. And if I'm not mistaken, I think the vast majority of these people chose as their subject for reproductive immunology pregnancy. Now, what button do I hit on this to make it move? That works. Okay. So it's pregnancy. Right or the left? Now, that's really a good answer. It's just as good for anything because I don't know my right from my left anyway. But they focused on pregnancy, and I admit turtles don't really do pregnancy, but we had to talk about turtles today. They're really cool, so I put that up there anyway. So mammalian people just live with it. But anyway, I think the reason that they chose pregnancy for their talks is because of this conundrum stated here. And that is the placenta and embryo are semi-allogenetic tissues. They have paternal antigens on their surface. These antigens should be recognized by the mother's immune system, and it should be rejected. Joe Kane in our laboratory found this quote from Dr. Peter Medawar. He said, "The human mind treats a new idea the same way the body treats a strange protein. It rejects it." You got to think that he probably just had a grant rejected from a study section when he wrote that, but it is pertinent to this presentation. Because you think about it, the day-to-day physiology of a mammal, pregnancy is a pretty new idea. And somehow physiology has figured out a way to overcome that urge to reject. Now, every one of those people I talk about that talked about reproductive immunology somewhere in the beginning of their talk paid homage to Sir Peter Bryan Medawar, because he's considered to be the father of immunology, and I'm doing the same here. So Dr. Medawar, he is considered the father of reproductive immunology. He won a Nobel Prize for the discovery of acquired immunological tolerance. He worked a great deal with graft rejection. It's interesting what you talk about how much we have now to work with and what little they used to have. You read the papers where him and McFarlane Burnett, you know, when you win the Nobel Prize, you get to have a Nature paper and stuff. And you see what little they had to work with and how fantastic their ideas were. This MHC stuff that I'm going to talk about, they were already getting on top of that. No molecular techniques to know that. This was just pure thinking and really being insightful. So sometimes, you know, the omics are fine, but sometimes you got to really sit down and think about the biology to advance things. But anyway, he won the Nobel Prize. He's the father of reproductive immunology, but he didn't work in reproductive biology, hardly at all. A little foray into free Martinism in cattle. But what he did do is he was the first to verbalize taking what he had learned and putting it against pregnancy, this conundrum. Here in 1953, he says, "How does the pregnant mother contrive to nourish within itself for many weeks or months a fetus that is an anagenically foreign body?" Then by 1961, he's getting a little more fine-tuned. He says, "Tolerance makes one think anew about the special relationship that holds between a mammalian mother and its unborn young. Yet it seems most unlikely that maternally induced tolerance should be a regular or well-defined occurrence," meaning you can tolerate this embryo, but you better not be tolerating every other thing out there. Our Dr. Raymond Berry was way ahead of his time when it comes to this kind of stuff. He did his interspecies transfers, as you just saw, with Bill said, between sheep and goats back in the 30s and 40s. None of those carried to term. They couldn't have known the mechanism why they didn't come to term at that time, because immunology was kind of in its infancy, and certainly there wasn't a field of reproductive immunology at that time. But the time, I don't know if Peter Medawar was ever aware of these studies. I really don't know. I haven't found any evidence of it. But I bet if he was, he would have been very interested in it. He wasn't scared to look at large species. That didn't bother him a bit. And that would have been a very interesting model for him. So, a significant barrier to maternal tolerance of placental fetal tissues is the class 1 major histocompatibility complex, the MHC. That's depicted here in this cartoon. It's a transmembrane protein. You have 3 globular motifs, and within those globular motifs, you have this peptide binding cleft. Now, the peptides that bind into this cleft actually come from your own cells, or come from the cytoplasm of your cells. The proteasome within the cells chops proteins up into peptides. These are then trafficked to the endoplasmic reticulum, where they're put into that binding cleft with the class 1 MHC. Then, like all proteins, you package them up through the Golgi secretory vesicles, and they place it out on the surface of the cell. Then your T cytotoxic cells come along with their T cell receptors, and they survey that antigen. If that antigen is foreign, they kill the cell. No negotiations there. You kill the cell. And in this way, your immune system is always checking out your own cells to see if something's wrong. If you've been invaded by a virus, you're going to be making foreign proteins, and it's going to kill you. If you've transformed into a cancer cell, you're going to be making foreign proteins, and it's going to kill the cell. That's going on. That's not even negotiable. And yet, when you think of that powerful system attacking anything that looks foreign, we see pregnancy becomes a major negotiation. On the immune system side, they say, I must protect against foreign invaders. But the placenta fetus says, I must survive to maintain the species population. So it's been successful in all these mammalian species, and I think the reason they've had success is kind of summarized here. Both the immune system and placentation are highly complex, variable from species to species, and I think the real important thing here is that they are both able to change and adapt rapidly on an evolutionary basis, and I think that's what's happened in each species. As placentation changes, the immune system adapts, and vice versa. Who's the first and who's the second? It may change all the time. So with that overview, I'd just like to start then with an overview of placentation in our farm species. I'm not going to belabor this. There's too many people in this room that understand these concepts better than I, but I will do this just as an introductory remarks. We've seen this one before, I see. So the attachment for implantation of the conceptus is an adhesion cascade. I hope I can do this as good as Nerve did. Okay, so, and that's depicted here. I drew this for the pig, but it's similar for the sheep. It's probably similar for the cow, and it's probably similar for the goat. Timing will be different. Some of the proteins and hormones will be a little bit different, but I could draw it out the same for those. The role of the conceptus in this cascade, and when I say conceptus, I mean the embryo, fetus, and the associated placental membranes. His role is to hatch from the zona pellucida, elongate, and then he's going to orient himself to the apical surface of the luminal epithelium of the endometrium. The role of the endometrium, then, is to downregulate these big anti-adhesive carbohydrates, like mucin 1 or MUC1. This then frees this interface here between the apical surface of the trophectoderm and the apical surface of the luminal epithelium for interactions between adhesion molecules. And in this case, we've got carbohydrate-lectin interactions. These are not strong interactions. They're pretty feeble, but they eventually then are replaced by much firmer attachment molecules, the ECM molecules and the integrin receptors. And the best described of these are osteopontin and its integrin receptors. Now these stages of orientation, apposition, and adhesion are depicted down below here. We've seen a lot of beautiful immunofluorescence and immunohistochemistry today, just amazing. Dr. Lee's lab is just it's amazing. What we see here is we've just done immunofluorescence staining for pCNA. So any of the off-colored nuclei, those cells are proliferating. Counterstain with DAPI, so we have a histological reference. So we can kind of see here we have the luminal epithelium of the uterus, the glands of the uterus, you have the stroma of the uterus, and here we have this conceptus up here. Not showing up so good because the lights are a little too bright for this kind of fluorescence, and he's still in the pre-contact stage at day 13. We call that free-floating. We all know that uterus isn't this wide in there. It's not free-floating, but he's not attached. Then by day 15, apposition, he looks attached, but we could flush that intact out of the uterine lumen if we wanted to. But by 20, there's firm adhesion of the luminal epithelium to the conceptus trophectoderm. Now to remind you of the cell types in placentation in our farm species, I'll go to this, for the uterus, I'll go to this cartoon provided by Dr. Rodney Geisert. We have a simple columnar epithelium, luminal epithelium, that lines the lumen. Invaginating down from that into the connective tissue is the glandular epithelium. The connective tissue is the stroma with the stratum compactum, stratum spongiosum, and then outside of the uterine wall is the myometrium with its inner circular and outer longitudinal muscle layers, and then of course we have a parametrium mesothelium on the outside surface. Placentation is a close juxtaposition of the microcirculatory systems of the mother and fetus for exchange. Okay, right away, if you're foreign, placentation is going against what we would think you'd need to do if you want to hide from the immune system, because you're just putting yourself as close as you can to the mother's immune system. And that's shown here for the pig, where we have the vasculature of the endometrium supplying the luminal epithelium in blue, attached it to the chorionic epithelium, or you could call this the trophectoderm in the yellow. And then we have the allantois, which is the connective tissue of the placenta and its vasculature, and then lining the inside of the placenta is the allantoic epithelium. That's the allantoic cavity that's filled with the allantoic fluid, who Dr. Baser just loves allantoic fluid. No doubt about that. So if we get a nutrient from the endometrial vasculature across the allantoic vasculature, which would then go to the supply of the embryo or fetus, you have to take that substance across an endothelium, across a basement membrane, across connective tissue, across a basement membrane, across a luminal epithelium, across to the chorionic epithelium or trophectoderm, across a basement membrane, across a connective tissue, across another basement membrane, and across another endothelium. That's epithelial chorial placentation, the least invasive type of placentation. And that's kind of depicted here, where we can see these respective epithelia in red and these vasculatures, the endometrial and placental vasculatures. This is early enough where the uterine vasculature is far more advanced than the placental, but the placental will catch up later on. Macroscopically, placentation in the pig is defined as diffuse, meaning the entire uterine placental interface folds to maximize surface area to get nutrients from the endometrium across to the allantois. In our ruminants, it's cotyledonary, so you have these placental cotyledonary villi interdigitating with these uterine caruncular crypts to maximize surface area for transport between these two vasculatures. Microscopically, pigs and sheep are epithelial chorial and semi-epithelial chorial, respectively. And this is defined by what cell type or what tissue in the uterus is next to the remaining chorion. So in the case of epithelial choria, it's the luminal epithelium, and I just described that for you. If you look at the ruminants, there's some degree of fusion, and it's different from each ruminant, between the chorionic epithelium and the luminal epithelium, and so we call that synepithelial chorial. I'm going to finally close these opening comments, and I'm going to get on my soapbox a little bit here, and say that epithelial chorial and synepithelial chorial placentation are not evolutionarily primitive. We hear this all the time from people from work with mice and people who work with humans. It's just not the case. Diffuse and cotyledonary placentation are late events, evolutionary speaking, and epithelial chorial placentation is depicted here for the pig and cow are also late evolutionary events. We are always told these models are too simplistic to be relevant. I argue that's no way that's true. They're relevant, and they're relevant to the comparative placentation and what we can learn about placentation across species. I think all the species have something to say and teach us about every other species, including humans. So with that, let's get to some data. And we have performed some studies to examine the expression and regulation of class 1 MHC during pregnancy in the uterus and placenta of sheep and pigs. And I remind you, the synepithelial chorial placentation is relatively non-invasive. And to put the cart ahead of the horse, essentially their strategy is the placenta defends itself behind a wall of non-expression of MHC from these invading immune armies. In sheep, the uterine lily and conceptus trophectoderm does not express class 1 MHC or beta-2 microglobulin proteins, simply meaning that the immune system is blind at that interface. This is work performed by Dr. Young-Suk Choi and myself a long time ago for my postdoc years. He simply looked at total RNA during the ester cycle in pregnancy for MHC class 1 and beta-2 microglobulin in the uterus. And what he found was a major increase in class 1 MHC and beta-2 microglobulin in the endometrium during the peri-implantation period. But this is not a story of total gene expression, it's a story of where the gene is expressed. Because when I performed the immunofluorescence data, And I'm sure Bob remembers sitting and helping me capture these in the dark. People used to talk about us. We sat in the dark alone a lot on the weekends. No hanky-panky. Bob wanted to, but I wouldn't let him do it. I like Becky too much. Anyway, at day 10 of pregnancy, that's before implantation, the lumen epitheliums express class 1 MHC. But by day 14, when implantation is occurring, it downregulates in the lumen epithelium and upregulates in the stroma, and by day 20, it's highly expressed in the uterine stroma. The same pattern can be seen for beta-2 microglobulin. Also note that neither beta-2 microglobulin or MHC class 1 are expressed by the conceptus trophectoder. Young-Suk then looked at the mRNA expression. And these are different days of pregnancy. This is for mRNA, so it's in situ hybridization, corresponding bright field and dark field images. You look in the dark field, where the white is, that's the cell type specific presence of the mRNA. And he saw the same pattern I saw with the protein. It upregulates in the uterine stroma during the peri-implantation period. And when you look closely at the conceptuses, the MHC class 1 does not have mRNA in the trophectoderm. Beta-2 microglobulin does, but remember, beta-2 microglobulin has other roles other than MHC, and it isn't going to elicit any kind of immune response if it isn't in the context of MHC. Another interesting thing here is these were overexposed. We weren't perfect on this, we overdid it, but this remaining luminal epithelium and shallow glandular epithelium is not expressing, but we have high expression in the underlying stroma. So we asked the question, what's upregulating these genes in the uterine stroma? And to do that, we use a model that we use till today. We took ewes and we overectomized them, we gave them progesterone, and we gave them progesterone plus interferon tau. And interferon tau upregulated both of these genes in the uterine stroma, and I could also show that on the protein level. So, class 1 MHC and beta-2 microglobulin are classic interferon-stimulated genes that increase in the stroma but are not increased in the uterine luminal epithelium. Also note that this stromal upregulation is not just fibroblasts. The vasculature is also expressing class 1 MHC. That was the end of the story for quite a while. But then Joe Kane in the laboratory has just recently taken this project up. He's going to look at class 1 MHC during later stages of pregnancy in the sheep. And this is preliminary data from way 110 of gestation. It's a placentome. These areas with the blue nuclei and the immunostaining, those are the caruncular crypts. The black areas are the cotyledonary villi. And you can see you maintain vascular expression of class 1 MHC in the caruncular crypts through day 110 of gestation. It'll be really interesting to look what happens between the peri-implantation period and day 110 in intercaruncular versus intercaruncular regions. And then we really want to look at placentomes just close to parturition, because we know that in cattle, MHC class 1 upregulates in those placentomes, presumably had form of inflammatory response to split cotyledons from caruncles for parturition. We've also looked in the pig, and again, I remind you, pigs are epithelial chorioplacentation, even less invasive than sheep. They use the same strategy as the sheep. They sit and defend themselves against a wall of non-expression. Margaret Joyce in the laboratory, she looked at the swine leukocyte antigens, which is the MHC in pigs and beta-2 microglobulin across the estrous cycle and pregnancy. She looked at SLAs 1 through 3, which are the classical SLAs, and then 6, 7, and 8, which are the non-classical ones. All of these genes increase during the peri-implantation period by total mRNA. But when we looked by immunofluorescent staining, it was a pattern like we saw with the sheep. It's highly expressed in luminal epithelium at day 12. That's before attachment for implantation in the pig. But by day 15, which now we are starting to attach for implantation, it downregulates in the luminal epithelium, upregulates in the stroma. It's quite a dramatic difference between day 15 of the cycle and day 15 of the pregnancy. And then that's maintained throughout about day 60, when you almost can't see these interferon-stimulated genes anymore. Margaret, of course, did the obligatory RNA localization. That's a lot of data to look at. Just focus here. Day 12 of pregnancy in the luminal epithelium. Day 15, it's moved into the stroma, and that's maintained for some period of time in pregnancy. And each these other ones, SLA 6, 7, and 8, show the same patterns, but they're at lower levels of expression. And I just reiterate what we're seeing during the peri-implantation period. downregulation of mRNA and protein in the luminal epithelium, upregulation in the stroma. So we asked, what is upregulating these genes in the stroma? Well, it's an gene in the pig. And we knew the pig they also produce some interferon delta, lots of interferon gamma. That's just after interleukin-1 beta. So we took conceptive secretory proteins from day 17 proteins. We loaded up Immunology, those conceptive secretory proteins into the uterine lumen of cyclic pigs, and this is a model I'll talk a little more detail in later in this presentation. But in a nutshell, what we saw is if you just did serum proteins, you saw what you saw in the ester cycle. But if you infuse these conceptive secretory proteins, it upregulated in the stroma, and you can see that by the protein level as well, and you can note here very easily that it's also expressed in the endothelium. So apparently, it is also MHC class 1 and beta-2 microglobulin are interferon-stimulated genes in the pig as well. So with that, enough of this discussion about our species, placentation hides from the immune system. And let's start to talk about the fact that our species placentation actually embraces and uses the immune system during gestation. And this was an argument and that was very effectively and forcefully made by Dr. Gilmore, I think one of the best Berry lectures we ever had, where he argued just a few years ago that immunological responses at the receptive maternal feed interface are not simply suppressed, but are instead highly dynamic. I drove Dr. Moore from the airport and to the airport, and on the way back to the airport, we were talking so darn much I missed all the turns. I had to turn around. Honest to God, I missed the turns. He was quite a talker, but he was really fun. So I'll begin with just some recent data that's generated by Dr. Hee-Wan So. He's doing something very simple here. He's just doing immunofluorescence staining for CD45. He's picking up about all of the mature immune cells in the environment. And then he's looking at intercaruncular and caruncular tissues from days 30 to 60 of gestation. And clearly, there's plenty of immune cells in the uterus during pregnancy, particularly in caruncular tissues. And look at all these immune cells in the caruncular crypts. And these are not static populations of immune cells, because work done several years ago, led by J.J. Manus. I don't know if you even remember J.J., do you? Remember J.J. Manus? We co-localized L-selectin and glycam-1 in placentomes of sheep. Now this L-selectin, that's an adhesion molecule on leukocytes that leukocytes use to adhere to carbohydrates expressed on the endothelial cells. They attach to endothelial cells, that stops them in the blood flow, and then it allows them to extravasate or move from the bloodstream into tissues. So when we co-localized L-selectin with glycam-1, we could easily see these immune cells that were localized within the blood vessels, attached to the blood vessels, and we know they're attached because then we co-localized von Willebrand factor with L-selectin. Von Willebrand factor is expressed by the endothelial cells themselves, and they're clearly attached to that endothelium. So we have immune cells moving in and out of these caruncular crypts. Recently, we published a paper, I think it was Nerve said something a little bit about this, that challenges the current dogma for how the syncytia forms at the uterine placental interface of sheep. And this is a dogma that's been out there a long time, and just a few years ago, I drew this cartoon to summarize it in a review paper. Trophectoderm cells fuse to form binucleus cells. They invade down into the luminal epithelium, fuse with a single luminal epithelial cell to form a trinucleate fetal-maternal cell. Then more binucleate cells will invade. They can either bind with a single luminal epithelial cell, with these trinucleus cell, or with these growing syncytial plaques. And eventually what you get are these big plaques of syncytia that are both of uterine luminal epithelium and conceptus trophectoderm nature. And that's depicted here by staining with the PAGs that we heard a little about earlier today. We challenge that dogma somewhat. We do believe that there are trophoblast giant cells forming in the trophectoderm layer. They do invade down into luminal epithelium, but we think when they get there, they're engulfing luminal epithelial cells that are undergoing apoptosis. And then they carry those apoptotic luminal epithelial cells down into the stroma where immune cells phagocytize and eliminate the cells from the environment. Then you have further invasion of these giant cells, lateral fusion, and the syncytial layer then turns out to be entirely of placental origin. That looks just like a human. So our evidence for this, okay, is follows. Okay, so this is immunofluorescence staining for E-cadherin. It only is expressed by luminal epithelial cells. And it's, this is the lumen. We don't have a conceptus in this picture. And the first thing you note right away is this syncytial process in the sheep is patchwork. This region is not undergoing syncytialization. That's a normal columnar epithelium. But this area is. You can see the degradation that's going on there. And when you look at the stroma beneath these regions, if you're not undergoing syncytialization, there's no immunostaining down in the stroma. But if you are undergoing syncytialization, we now have E-cadherin positive cells down in the stroma. This is something that's not supposed to happen. You're not supposed to have epithelial cells invading into the uterine wall. You see these gaps here? We think that's because they moved those cells down into the stroma. Further, these E-cadherin positive epithelial cells within the stroma are undergoing apoptosis. Again, we got a region here where one part of the uterus is not syncytializing, the other part is syncytializing. And when you do immunofluorescence staining for tunnel, which looks for apoptosis, there's increased number of apoptotic cells there. But more importantly, when you co-localize E-cadherin with tunnel, every one of these E-cadherin positive cells are undergoing apoptosis. I didn't believe he won on this. I honestly didn't when he showed me this. I said, it can't be that perfect. And it is. They're all undergoing apoptosis. So they're not long for this world. We're getting rid of the luminal epithelium. Finally, immune cells are enriched near apoptotic epithelial cells. Here's another region where it's undergoing apoptosis, not apoptosis, syncytialization. Another region where you're not undergoing syncytialization. You can see a lot more immune cells in the region undergoing syncytialization. And if you co-localize tunnel with CD45, you can see these apoptotic epithelial cells are completely surrounded by immune cells, giving them ample opportunity to phagocytize and eliminate them from the environment. Now, I'm going to turn to something that I know you all knew I would turn to, my old friend osteopontin. I guess the group's getting young enough now they don't realize that you're supposed to laugh when I say osteopontin. I think I gave a talk before, and Joe Roche said, "You love osteopontin," and I told him, "No, actually, osteopontin loves me. He keeps popping up on me." But anyway, back in 2000, Dr. Harvey Cantor's laboratory published a paper in Science where he said that osteopontin is an early component of type 1 cell-mediated immunity. One of its early names before they knew it was osteopontin was early T-cell activation factor 1. Now, naive T helper cells can either become T helper 1 cells or T helper cells depending on the cytokines in the environment. T helper 1 cells favor cell-mediated inflammatory responses. T helper 2 cells favor humoral or anti-inflammatory responses. Interleukin 12 induces the transformation to T helper 1 cells. Interleukin 10 inhibits the transformation into T helper 1 cells. And what Harvey Cantor's he showed that osteopontin increased the effect of interleukin 12 and decreased the osteopontin in the environment. This is an inflammatory environment. The problem is we, for years, hypothesized, have claimed, absolutely knew, that pregnancy required an anti-inflammatory environment. And that just can't be the case because osteopontin is highly expressed at the uniplacental interface of many species. Now, I'm just going to show you the expression of osteopontin in the sheep, pig, and mouse. So when I first came to the laboratory of Dr. Fuller-Baser for my postdoctoral studies, he assigned me the job of looking at osteopontin in the uterus of the pregnant sheep. It's the best assignment I ever got in my life. Look at that data. It holds up beautifully to this day. I did that. It's expressed in the glandular epithelium throughout pregnancy. It's expressed a lot of other places, isn't it, Bob? But that's what we saw then. It's hard to see anything else when you see expression like that. It's actually upregulated earlier, about day 15 of gestation. That was in an earlier paper. We thought this would be upregulated by interferon tau. That was the hypothesis in the grant. Interferon tau has nothing to do with osteopontin. But if you overectomize sheep and give them progesterone, or give them progesterone with ZK, a progesterone receptor antagonist, the progesterone upregulates osteopontin in the glandular epithelium, like you see during pregnancy, and blocking the progesterone effects ablates this. And this results in osteopontin being secreted into the uterine lumen, because this Western showing increased amounts of osteopontin protein in the uterine lumen of day 15 pregnant over cyclic pigs, sheep. It's hard to keep track of these different species. And then that results then in osteopontin at the apical surface of the luminal epithelium and the conceptus trophectoderm. Note here, there is no osteopontin mRNA in luminal epithelium, and I don't show you the data, but there's no osteopontin mRNA in this conceptus trophectoderm at this period of pregnancy. So what happens, it's upregulated by progesterone in the glands, it's made protein, is secreted into the uterine lumen, and that's binding back to the luminal epithelium and conceptus trophectoderm, and this is a bridging ligand between ligands for the attachment phase of implantation. This promising data that we were getting in the laboratory at the time caused me and Hak Young Ka at the time, we went a little bit rogue. We proposed to look at osteopontin in the pig. We had no funding for this. There was no grants to look at osteopontin in the pig, but as always, Dr. Baser was totally supportive. He says, we'll find the money somewhere, although he had a lot of money at the time anyway. So. But we went ahead with this, and Jane Garlow ran the in situ hybridization, and she found osteopontin increased in the luminal epithelium between days 12 and 15, really right about day 13 of gestation. It was maintained there throughout gestation. It upregulates in the glands between days 30 and 35 of gestation. Now, those of us who work with the pigs and we see an upregulation between 12 and 15, we're thinking estrogen immediately. The pregnancy recognition signal in pigs is estrogen secreted by the conceptus. So Frankie White in the laboratory then compared the expression of osteopontin in day 15 cyclic, day 15 pregnant, and day 15 cyclic pigs that were then intramuscularly injected with estrogen to make them pseudo-pregnant. What he found is there's no expression during the cycle. We already knew that. There's high expression of osteopontin in the luminal epithelium, but just adjacent to the implanting conceptus. But if you give cyclic pigs estrogen, it globally upregulates the osteopontin throughout the luminal epithelium, albeit at lower levels than what you see at pregnancy. So it's pretty clear that the conceptus secretes estrogen, which then acts on the adjacent endometrium to increase osteopontin in the luminal epithelium, which is an adhesion molecule for that implantation process. Now, expression of osteopontin in the uterus and placenta, in the luminal epithelium and placenta is not the only place we see osteopontin expressed in the uterine placental environment. Here is estrous cycle of a pig in the endometrium, and we see high populations of immune cells expressing osteopontin mRNA. And I didn't mention that earlier, but we saw immune cells expressing osteopontin mRNA pre-implantation in the pregnant pig uterus. We've never followed this work up. It's probably something we should do. But we propose that this may be the way the uterus is prophylactically preparing for the possible introduction of pathogenic bacteria or something during the mating process. That's not the only species that has immune cells in the pregnant endometrium that express osteopontin. Frankie White did mRNA and protein in the non-decidualized, so this is the early pregnant mouse uterus, and he has patterns that look like their immune cells. He co-localized with CD45, osteopontin CD45. They are immune cells. Then he co-localized osteopontin, remember another name for that's SPP1, with marker to macrophages, and we have macrophages in the non-decidualized pregnant endometrium mice. And just this year, Avery Kramer and David Erickson confirmed that osteopontin is expressed by these famous decidual natural killer cells of mice. Several of the previous Berry speakers have talked about these decidual natural killer cells. They have a massive place in the decidua of pregnant mice, and they continue to confound reproductive immunologists because nobody knows exactly what their biology is. I could spend all day just talking about those guys. So now I'd like to finish this talk today with a discussion about the pro-inflammatory cytokine interferon gamma. Interferon gamma can stimulate the inflammatory effects of T helper 1. We've seen interferon gamma is a very popular gene to look at whenever you're doing cytokine arrays. And it will inhibit the formation of T helper 2 cells. And this is really a timely discussion today, because just last week, Dr. Rodney Geisert at the University of Missouri published in BOR his paper where he described CRISPR-Cas knockout of interferon gamma in pig conceptuses and its effect on pregnancy. And as you see by the title, he says conceptus interferon gamma is essential for establishment of pregnancy in the pig. That's fine and good, but I'm more interested really in this line here where he says ablation of conceptus interferon gamma production resulted in the reduction of stromal CD3 positive T cells. And I'm going to use the rest of this presentation to tell you why we're absolutely not at all surprised by what his results are. In fact, they confirm what we already knew. So this is work that began with Margaret Joyce. It's ended with Brian McClendon. It's not ended, but that's the latest stuff we have. We see that as the conceptuses elongate, they make high amounts of interferon gamma, mRNA, and protein. Again, we have in situ hybridization here where we're looking at the film so we can see the entire uterine wall. These are serial sections that have been probed for interferon gamma and STAT1, and STAT1 is a classic interferon-stimulated gene. Interferon gamma is expressed by the conceptus, STAT1 by the uterine wall, but only next to where the conceptus is implanting, not in other sites, strongly suggesting that interferon gamma acts on the uterine wall at sites of implantation to upregulate this interferon-stimulated gene. So we thought about that. As I told you earlier, SLAs are interferon-stimulated genes, so we thought maybe interferon gamma is having an effect on the immune system in the endometrium at implantation sites. And just a cursory look for M1 inflammatory macrophages, some markers for these, nitric oxide synthase 2 and arginase, shows that this is really a potential, that it could be an inflammatory environment in the endometrium. And of course, interferon gamma is an inflammatory cytokine. So we returned to He Hwan So, and he did some immunofluorescence staining using CD45, and there is an increase in total immune cells during day 15 and 18. He then did tunnel staining, and he found a modest increase at day 15. Tunnel is for apoptosis. Apoptosis is a hallmark for inflammation, and when you talk about inflammation, T cells become a topic of interest. So we did immunofluorescence staining for CD3, that same marker Rod mentioned in his abstract, and indeed it increases in the endometrium on day 15, 18, and 20 of gestation. But more importantly, many of these T cells, when you co-localize with PCNA, are proliferating. Now, a proliferating T cell is an activated T cell. And to activate a T cell, that T cell has to use its T cell receptor to recognize antigen in the context of MHC expressed by the antigen-presenting cell, but it must at the same time recognize a co-stimulatory molecule expressed by the APC. If it just recognizes MHC or just recognizes co-stimulatory molecule, you won't activate that T cell. Here's just some of the list of some of these co-stimulatory pathway families. Here's the B7 CD28 family. Best described of these co-stimulatory families for activating T cells. At the right here, we have the T cell receptors on T cells, and on the left, we have the ligands and antigen-presenting cells. And we chose to look at PDL1, CD80, CD86, and ICOS-LG for ligands on APCs, and PDCD1, CD28, CTLA4, and ICOS for receptors on T cells. So during the normal early peri-implantation period, when we looked at ligands on APCs, only CD80 was upregulated. But when we looked for receptors on T cells, every marker we looked at was upregulated in the endometrium during the peri-implantation period. So we asked, is this interferon gamma that's causing this? And to do that, we went to this mini osmotic pumps again. We installed mini osmotic pumps and infused interferon gamma protein into the uterine lumen of pseudopregnant pigs. So we checked for estrus. Estrus was day 0. Days 11 through 14, we gave intramuscular injections of estrogen to make them pseudopregnant. And then on day 13, easy for me to say, we did surgery. And in that surgery, we ligated the uterine horn proximal to the body. Then we took a catheter, threaded it into the oviduct and through the uterotubal junction. We connected this mini osmotic pump to the catheter filled with interferon gamma, and then we secured it within the ovarian bursa. We then collected samples at day 18 of pregnancy. What we found is if you infuse interferon gamma into pseudopregnant pigs, you have no effect on the ligands for the antigen-presenting cells. But when you infuse interferon gamma to pseudopregnant pigs, the receptors on T cells, you increase PDCD1 and ICOS, and there's a trend to increase CTLA4. This means we've just simply got more T cells in this environment, so we looked for chemokines that might be recruiting T cells into that environment. And we look at the CXCL9 through 11, and all of them dramatically increased during the peri-implantation period in the endometrium. And if you infuse interferon gamma into pseudopregnant pigs, you increase CXCL9 and tend towards increasing CXCL11. Finally, Brian did fluorescence-activated cell sorting to determine the percentages of the subpopulations of endometrial T cells during the peri-implantation period. And what he found is most of these cells are those very important T helper cells, CD4 positive. But there are other subpopulations. You have the CD8 C-cytotoxic cells, you have CD4 positive, CD25 positive T regulatory cells, and you have some CD4 positive, CD8 positive naive T cells. So with that, I want to acknowledge our interdisciplinary faculty of reproductive biology. After watching that set of fantastic talks today, this is a very healthy group that we've got here. Not very many professors here, but we have very, very healthy trainees. I want to acknowledge our funding from the USDA. They've been very kind to us. I want to acknowledge a group of people that are right now actively collaborating on the College of Veterinary Medicine and Biological Sciences and in the Department of Animal Science. This is our current laboratory in front of our, well, not brand new vet school, but still pretty new building. And with that, I'd be happy to take any questions or entertain any discussions. Mm-hmm.
So you should often was expressed in T cells. So these T cells go for
Boy, have we got a lot of data on that. We know that osteopontin upregulates the incorporation of endothelial progenitor cells into the vasculature. In the pig, we've got, I didn't show that data because it happens after day 25, but osteopontin is expressed in the placenta itself, and the protein localizes in globs around the placental vasculature. So absolutely, I'm absolutely positive. And then when we knock down beta-3 integrin, what dropped down? Osteopontin in the conceptus and nitric oxide synthase. So I'm almost positive osteopontin has a role. And there's a lot of literature from the past as well on that. No, we just.
Okay, that was it. Was there M2 and was the change?
We didn't go that far. We just didn't go. We didn't go into the macrophages. I get asked that every single time. We haven't done it, but that would be an interesting way to study. Yeah. Well, you know, osteopontin, it was 2AR, ETA1, and osteopontin, and they never knew they were the same gene until they sequenced it and said, wow, we got the same thing. Oh, boy. There's a lot of literature on osteopontin in the cancers because osteopontin is a drama queen. If you're a metastatic cancer, that every microarray that comes up, it's one of the top genes that pops up. Grace in the lab has been looking at Spark, and it pops up too, and they tend to go against one another. So, I suspect, and I go to osteopontin meetings every 5, 6, 7 years, and it's so weird because I'm the only reproductive biologist. These people come from every other area of biology, So they really always like our talks because everything's so dramatic with our hormones and stuff. Things come on and off, and they have such lower levels of expression. So it's been a while since I looked at the cancer, but I've always thought that had something to do with osteopontin's ability to increase the vascularization of those cancers. That's what I think. But it's definitely a pro-inflammatory, it's pro-cancer growth. If you're metastatic, you're probably going to be expressing osteopontin. So he's a chameleon. He can be post-translationally modified many ways, and it changes his function. So that's why people at those osteopontin meetings always complain. Because they say, we can't get funding for this gene, because people just roll their eyes and say, now what is it doing? Honest to God, everybody has that problem. They say, come on, what doesn't it do? It's the problem you have with something that's, I think it's got more roles than fibronectin, personally. But
one of the characteristics of two proteins we talk about the enterin gamma and SPP1. You do not want to freeze and thaw because it will go away. A lot of people, including our lab, when I was started, we thought we had this protein related to the internal sequencing and gone. So yeah, they came from protein products.
that must have been one of those moments. Baser was in the shower and he's thinking, I keep losing this protein. I just read this paper that this protein is not stable, and that came up with the whole hypothesis. The only thing you were wrong on was it wasn't interferon tau. Although there's a review paper out there that I'm not going to say who says it is, but we have something back here.
Go ahead. All right. Thank you. So I just want to reiterate everything people say about the microscope skill tools. Earlier today, I said Dr. Prairie, 90 years ago or so, brought the first microscope into fundamental science. I'm pretty amazed and impressed. Ask questions.
I think Dr. Peter Medawar would be pretty amazed and excited about what we're doing.
So I want to thank Dr. Johnson. And so we have this platform from Physiology of Reproduction, Texas A&M University, Raymond Memorial Lecture, 2021. So thanks for a great talk. And congratulations.
Thanks.
I'd also like to reiterate what Dr. Johnson said. We have some great talks today. Hopefully next year we get back to the post-COVID normality and more posters and more talks. But anyway, we'll adjourn for this